All right, so let's get started today. So we are going into transcription and translation. Gene regulation we'll go over tomorrow. Okay, and just a reminder that tomorrow tomorrow there's a lecture quiz. I don't think there's any more lab quiz, so I could just delete that portion. Okay. All right. So let's get started on today's lecture on transcription and translation. Before we start, you guys know transcription translation, right? Okay. So if you guys understood that, what I want you guys to do is I want you guys to discuss among each other. Maybe Nathan, Nicole, maybe you guys can sit over here. And then, let's see, Itimizo, join this table. And Sigavir, Valerie, Jacob, join the other table. I want 5 of you guys to discuss among each other what exactly is a central dogma, and what exactly is central dogma in science. Okay, and does the central dogma hold true? So go ahead and just take the next 2 minutes and discuss about that. All right, guys. So let's go over together. What exactly does it mean by central dogma? What is that? You guys heard that term before, right? So what is central dogma in science? Anyone volunteer? Anyone can shout out.
It's just a general idea that DNA is made to RNA and made to protein. It's still happening.
Right. It's a general idea from that DNA would turn to RNA, and RNA would turn to protein. And the question for you guys is, does this idea still hold true? What do you guys think? Does the central dogma idea still hold true? Any volunteer besides Nathan? Anybody here? From your past teachers or your past lessons? What do you guys think? Does it still hold true? You don't know? Yes? Well, I mean, it makes sense, right? I mean, DNA would turn to RNA, and that's through something called transcription. And then from RNA, you turn that to protein, right? And that would be translation, right? So the question is, does it still hold true? I mean, if you think about it through an analogy, let's just say you purchase a cookbook, okay? This cookbook you could transcribe by writing down the protocol of what's found in the book, right, onto a separate piece of paper, so you don't burn the whole book if you accidentally burn it. So you just follow that paper. That would be transcription. And then from that piece of paper or your notes, you're going to follow the ingredients or protocol to make your meal. And that'll be translation. And that makes sense. But could you do the opposite, where you cook the meal first, and then later you put it on your paper? So cook the meal first, then put in your paper, and then from this paper you make a book. Is that still something you could do, or you cannot? Can you vice versa that? Right?
Yeah.
Think about it. Analogy. Can you do that if you were to, after you cook and you finish your meal, you decided to rewrite that protocol how you made it, and then at the end you decided to make a book? Is that something possible? Yes, it is, right? It is possible. So the idea of the central dogma, back then, in about 1980s, actually this term was thrown around in 1950s, the central dogma, scientists didn't realize that this mechanism could go backward. They only believe it goes forward, which is why they call it the central dogma, the dogma of science. And dogma is a terminology usually found in religion, where it has to be it, cannot change, right? But in reality, we did found evidence that it could be reversed. For example, a big evidence that most of you guys already know is reverse transcriptase, turning RNA back into DNA, right? That's a huge reversal. What about protein into RNA? Well, there's possibility that could happen, and that goes to the time where scientists. This still go back to the beginning of time. In the beginning of time, did the first organism have DNA, or did the first organism have protein, or have RNA? So it goes back to that time. If the first organism only had protein to start out with, then how did that protein become RNA and then became DNA? Right? So if you were to talk about all of those, it is possible to go reverse. So again, just a summary, what I just mentioned there. Central dogma only means going from DNA to RNA to protein. But nowadays, we do know there is new discovery that things can actually go backwards, such as RNA to DNA through reverse transcriptase, right? And there's a possibility that protein is started out in the beginning of time, is the first trait to be passed on from generation to generation, and eventually that leads off to, eventually becomes RNA and eventually become DNA. Okay? All right. And not to mention, remember that this is telling us DNA has to become RNA and has to become protein. But in reality, they don't have to be. RNA itself has its own function, and DNA itself has its own function. So for example, here are some examples. DNA aptamers are basically DNA, but acts as a receptor or an inhibitor. So a cell could actually express DNA to just inhibit another cell. That's crazy that it could do that. And then we also have regulatory RNA, and we express this naturally, right? A lot of our non-coding RNAs, they don't code for anything. That's because they have in itself their own function. They could be microRNA, small interfering RNA, or even riboswitch. So RNA and DNA have their own unique function as well. Here's another big example, tRNA. That in itself is an RNA that acts as an enzyme. That's crazy. So in other words, it doesn't need to follow the whole trend from DNA to RNA to protein. It could just stay DNA and has its own function, or it could become RNA and stays RNA, right? So what I'm trying to tell you here is that in regards to central dogma, back then when they coined the term central dogma, it sounds really good back then, but nowadays we know more. And there are a lot of things that basically shows central dogma is not really fully true. Although it's good to understand the fundamental though. All right. So where exactly does transcription and translation takes place? Welcome back. So where exactly does it take place? Well, in regards to prokaryotic cells versus eukaryotic cells, they take place in two different locations. Well, in prokaryotic cells, both transcription and translation occurs in the cytoplasm. Okay? And they occurs simultaneously. So what does that mean by simultaneously? Let's just say you have your DNA right here. My arm is a DNA. And it is transcribing an RNA. Pull this out. I'll see what you. And let's just say it transcribed an RNA. So here's your RNA being transcribed from the DNA. Okay? And at the same time as being transcribed, not done yet, it's still a long way to go. As at the same time as being transcribed, a protein comes in and start translating it. So it's being transcribed and translated all at the same time. That's what it means by simultaneous. So as the RNA is being built, this RNA, even though it's not loosened out, it's still being built, protein is already being made at the same time it's being made. That's crazy. So that's what happened in prokaryotic cells. On the other hand, eukaryotic cells, we have something called the nucleus. Right? This organelle, the nucleus, holds our DNA. So in this case, our DNA transcription, which turns to RNA, occurs in the nucleus. And then in order for translation to occur, that RNA has to leave the nucleus, right? So transcription occurs in the nucleus, which is DNA to RNA, and then the RNA will leave into the cytoplasm, and this is where translation occurs. So in other words, translation and transcription in eukaryotic cells occurs in different locations. Now, something very interesting to also note as well, in regards to prokaryotic cells, about 80%, if not more, of their DNA, overall DNA, is actually transcribed to something functional or feasible, right? Something that's actually work. On the other hand, in eukaryotic cells like us, human, less than 8% of our DNA is actually transcribed to something functional. That's crazy. I mean, for bacteria, 80% of the DNA is transcribed, but they only make like maybe 500 genes. While human, only 8% of it is transcribed, but we make more than 25,000 different genes. Holy cow. How's that even possible? We only express 8%. They express 80, but we express more? How's that even possible? So what I want you guys to do now, take the next 2 to 3 minutes, discuss among your table, and discuss why only 8% of our DNA is transcribed, but we still express more than 25,000 different genes. All right,
So
so
go ahead.
any volunteers? I want to hear some thoughts. This is all theoretical as well. I want to hear your thoughts. So what do you guys think? Only 8% of our DNA is transcribed, but if we still express a whole bunch of genes, how is that even possible? What do you guys think? Yes.
Parts of our DNA is just random code, so some of it's actually not being we don't actually use it, and a lot of there's also like parts of it where it's on the ends of the DNA, so it a lot of random coding so that it just protects the actual boundary.
Okay, so we have a bunch of random coded codes that protects our DNA. Cool. Any other suggestions that you guys think? Why in eukaryotic cells only 8% of our DNA is transcribed? Any other suggestions you guys have? Yes.
Introns and exons.
The introns and exons, that's a good one too. Very good. All right. Well, I want you guys to think about this, okay? Hopefully this kind of pop an image to your head a bit more. Do our brain cells express the same gene as our liver cells? What do you guys think? Does our liver cell express the same gene of our intestinal cells? How about our white blood cell? Do they express the same gene as our red blood cells? So hopefully that clicks a bit more. So go ahead and continue discussing a bit more why we have only 8% of our gene and not all of it. So go ahead and discuss about that. So let's go over this together. So only 8% of our gene is expressed. Well, there's a lot of factors, as mentioned before, that kind of hinted towards the answer. The reason why we only have 8% of our DNA that's only transcribed, there's several reasons. For one, I'm not sure you guys know this, but back in the days, just imagine billions of years ago, we used to be single-cell organism, and one of the worst enemies that we have to fight off all the time are viruses. And so over the billions of years fighting off against viruses, we developed mechanisms to lock in their DNA permanently to where they cannot be expressed. So if you do not know so far, nearly 20 to 40% of our genome, our genome, are ancient viral DNA that we completely conquered. They cannot be unlocked anymore. Actually, some of them are still alive. We call them transposon. That's an ancient virus, transposon. You guys heard that term before, right? But most of our gene, about 40%, it's our ancient virus that we locked in our genome, right? But that doesn't really account for the 92%. What's going on there? Well, the next factor is what you mentioned earlier. There's something called telomeres, and all of those, the ends of our DNA, always gets longer and longer and longer for some reason. And that's because there's an evolutionary advantage of having longer and longer ends. And that's because the longer ends we have, the less likely we undergo some form of DNA loss. So imagine your DNA doesn't keep losing the ends. That'd be dangerous. If we keep losing the ends, there's a chance that we may lose the important gene. So therefore, evolutionarily, we prefer having longer tail. Every time you pass on that gene, the ends of your chromosome gets longer and longer. That's because of preservation. So we just make a bunch of random letters to just continue preserving our genome. So naturally, over time, it will be longer. That's just to protect our own genome. And another one is, when scientists said only 8% of our gene, they're referring to one cell. That one particular cell. Maybe they're looking at white blood cell and they're looking at that. Only that guy has 8% gene. But if you were to account for every single other cells, it's going to be more. The reason why I said 8% of our gene, it just means 8% of the gene of this cell is active. Maybe if you look at the liver cell, it's another set of 8, not the same one, right? Maybe you look at a different cell, like a brain cell, maybe 16% is active, right? So in other words, every single cell from the same DNA strand, they will express different genes at different locations. So our genome is very, very complicated. The good news for you guys, I'm not going to be testing on that. I'm mainly going to be testing on you the fundamentals like central dogma and where does translation and transcription occurs. In regards to this information, that's for you guys. Just fun fact. Okay? All right. Because it gets kind of complicated if you think about it. Okay? But anyways, let's talk more about the central dogma, going back. And what I want you guys to do, I want you guys to really understand how to do transcription and then how to do translation. So in regards to transcription, you guys understand complements, right? A always will become T, right? And then C become G. You guys know those fundamentals. And then in regards to RNA, A becomes U, right? U becomes A. Or A becomes, A becomes U. And then T becomes A, right? And then C and G. So what I want you guys to do is really understand how transcription and translation works. So here it is. This is a parental DNA. After you transcribe it, everything is complement. So here's A. AAC becomes UUG, right? And here GAC becomes CUG. And then TAC becomes AUG, and so forth. Okay, this is just complements, very simple. And I really want you guys to, when you guys have this kind of questions on your quiz or exam, I really want you guys to write it out. Don't rush it. Don't skip points. Just do the whole thing. Because there are a lot of students that always makes mistakes because they think there's a shortcut. There's no shortcut. Well, actually, maybe if you find TAC, but that's later on. Okay, but there's no shortcut. Just draw it all out. All right. The next thing I want you guys to recognize is turning mRNA into amino acids. This is through translation. Now, a lot of students, the biggest mistake is that they always start from the beginning. That's the big mistake anyone can make. So, what do we mean by start from the beginning? It's like, oh, look at that UUG. So let's take a look at U, and then G. Oh, okay, great. It's going to be a leucine. It's a nonpolar. Awesome. Right? But in reality, when you begin translation, you have to look for the key letters. AUG. If you don't find AUG, then that's going to be an issue, right? Because this is where you have to, this is how translation works. You have to find a start codon. And that's a very important key. So it doesn't matter how many sequences are there in front of you. If you cannot start translation without AUG. Once you found AUG, that's when you start. So AUG stands for methionine or start, right here. And afterwards you go to the next one. Let's go next one together. AGU. So you're gonna start off using this whole entire DNA table. A is on first, is on the left, followed by second, followed by third. Okay? So A, where is it? Here it is, A. And then there's GU, okay? G. Here's G, so it has to be somewhere here in this box. G, and now you look for U. AGU, serine. Okay? Serine, that's mean S. Next is ACU. A, somewhere in this row. C, it's going to be here in this box. And then U, right here, ACU, threonine. Next one is CGU. G is down here, somewhere in this row. C, GC in this box. And then there's going to be U, alanine, CGU alanine. And then here ACU, A, C, U, threonine. And then finally UAA. So you're going to have U, A, A. You only stop whenever you see the letter stop. In this case, UAA stands for stop. So now what you did, you transcribed the word start or methionine, S-T-A-T, stop. Okay? So just a reminder, whenever you do transcription, make sure you write down the whole thing. Don't take shortcuts to transcribe it. And remember, A becomes U, okay? Don't forget that. And when you guys do translation, remember, don't start from the beginning. Look around for AUG. And remember, sometimes I roll out like this, 3 codon at a time to make it easy for you. But keep in mind, sometimes this A is over here, and UG is on the second, right here. So AUG. So keep that in mind as well. So it's not going to look beautiful like how I put it. I do this just to represent, so it's easier for you guys to follow. But keep in mind, your goal is to look for AUG no matter where it is in the DNA, in the RNA. But that being said, what I want you guys to do now, so I had food poisoning, guys. So what I need you guys to do now is, I'm also a super nerd. So for some reason, I wrote a letter, and your goal right now is to find me an antidote. But just to have fun with you guys, I just gonna write this whole sequence here. So your goal, you have 10 minutes. Your goal is to figure out where is my antidote. I have food poisoning, guys. Go. let's go over this together. So first thing is first, the shortcut is looking for, well, in my class, I'm just going to keep the DNA sequence easy, but just be aware if you do transfer out to a university, they may flip the script around. Like this could be 5 prime and down here it could be 3 prime. So in other words, you have to reverse this whole thing again. So if this was 5 prime over here for some reason, then make sure you rewrite it to where you rewrite everything back to where it's 3 prime to 5 prime for DNA. Okay? So it's very important, but for me, I keep it simple. I don't want to overcomplicate it. So I'm going to do 3 to 5 prime here. Okay? So all you need to do now is look for, the shortcut is look for TAC. So you look around if there's any TAC around. And once you found TAC, oh, here it is. This is where I begin my transcription translation. So in this case, TAC stands for AUG, which is the start codon, right? So that's how it kind of works. So tic-tac-toe. So here it is, tic-tac. So we'll start from there, and then we start transcribing. Which we'll start transcribing everything here. And once you transcribe all of it, the answer you should get is SEAT. SEAT. That's the answer you get. Now, if you for some reason forgot about this and you start translating it right away, you're going to skip the word try again. I did that. It's kind of cool, right? I did that. So if you start from the beginning and you start transcribing it from the beginning, rather than starting from here, you're going to get the word try again. All right. Cool. So any question in regards to transcription translation? Yes.
So if it really was written 5 times to 3 times, you said flip it and just rewrite it? Or
Yeah, you can rewrite it. So if this is 5 and down here is 3, you're basically going to rewrite it to where it's going to be, you're going to write down 3 A C A T T A A G. So you're basically writing it backward. Just rewrite it for yourself. Okay? But there will be other classes if you plan to transfer to any university and you're taking genetic. That's you, Nika, and Ian, right? You're going to go to UCI. They will do that to you. And Nathan, if you're taking genetic there, they'll do that to you. So be careful. They love doing that. All right. Okay, so first thing is first, that was transcription and translation. Hopefully it wasn't too bad. Make sure you guys understand how to use this. On our exam, the written portion, you guys will have this, something like this again. And then, of course, because this is biochem, I take it to the next level. And after you found out what protein it makes, you have to draw that protein out. And tell me its function. Right? All right. So that's going to be the written portion. So that's a little hint there. Well, I mean, you guys can have time to practice that on Wednesday. All right. So let's go into transcription now. How exactly does transcription work? So I'm going to begin off with transcription from prokaryotic cells. And then I'm going to go briefly into eukaryotic cells. Okay? And then we'll move on from there. So in regards to prokaryotic cells, there's 3 main steps in regards to transcription, which is from DNA to RNA. So this is the DNA right here. Okay? This is RNA polymerase. So it uses RNA polymerase, okay? So first step is initiation. Initiation is when the RNA polymerase bind onto the promoter. That's pretty much it. That's pretty easy, right? And then elongation is when it opens the promoter and start transcribing the RNA. That's elongation until it's done. And then termination is when the RNA terminates and it just kind of leaves, it's just kind of break apart from the DNA. That would be termination. Okay? So to make it simple, I mean, it sounds simple, but some of you guys love to overcomplicate stuff. So let me just animate out for you guys. So please do not overcomplicate it. This is where it always gets hard if someone's trying to overcomplicate it. All right, you guys ready? Initiation. So the RNA polymerase binds to the promoter. You guys get there? Cool. Elongation. It creates the RNA. You guys good? And then termination is when the RNA branch out and just kind of dislocate. Is good? All right. I don't know how it's that hard, but when I was teaching micro, it was insane how students do bad on it. But just don't overcomplicate. It's that easy. But let me add a bit more complexity to it, though. How exactly does it terminate in the first place? It just sounds so easy. So how exactly does it terminate? There's two methods to terminate. Okay? One method is at the end. Let's just say this is the end tail. The sequence, and we'll call it self-termination mechanism. During self-termination mechanism, all it does is the end of the RNA. Imagine this mRNA here is super long. Okay? The end of mRNA, it will make a loop. It will create a loop. Okay? Just a loop. When it creates this kind of loop, it causes a very strong tension. All right? And that tension basically rip off from the DNA. It's almost like you're bending the DNA, and the DNA does not like to be bent. So having that loop like this cause that tension, and because this tension is so strong, it will rip off from the DNA. This is what we call self-termination. Okay? Because it causes the loop, and that loop causes tension. And the second mechanism, the last one you guys know, in regards to termination, it's called row-dependent terminator. Row-dependent terminator, it's not too bad. All it is just an enzyme that follows follows DNA polymerase. So imagine DNA polymerase is transcribing. There's going to be a row protein that's moving along and following the DNA polymerase. So as your RNA is being transcribed by the RNA polymerase, right? There's going to be another protein behind it called row protein. It's just going to follow it. And this row protein, all it does is looking around for a specific sequence. Once it finds that specific sequence, aka the terminator sequence, it will then cut. So this row protein has a scissor mechanism. So once it finds that particular sequence, it will then cut, and now the RNA is then cleaves out. Cool? So those are two mechanisms. One is the loop, which is called self-terminating, and the other one is through a protein that cuts when it finds a specific sequence near the end. And that's called row-dependent. All right. Any question about transcription? You guys solid? All right. And the last one I want you guys to know in regards to transcription is going to be the eukaryotic cells. Now, I don't want to overcomplicate this, but pretty much the main differences between eukaryotic cells and prokaryotic cells is that prokaryotic cells, they have something called transcription factor. Why is transcription factor so important when it comes to transcription? Well, imagine this is your DNA right here. Right here, okay? Now, imagine you have a bunch of promoters and a bunch of DNA everywhere. You have 3 billion strands of DNA, right? So this RNA polymerase could bind to anything, right? And start transcribing. Wouldn't that be dangerous if you accidentally transcribe a cancer gene? Or a anti-repair gene that stops you from repairing? Wouldn't that be dangerous if your brain cells start expressing cells from your muscles? Right? It will cause tumor. So in order to prevent that from happening, our own genome purposely nullify the DNA to where it's so insensitive. So in other words, enzymes like RNA polymerase cannot even recognize it's even there. It's almost like invisible. So in order for RNA polymerase to even recognize that our DNA is even there has to be another feature. We call this transcription factor. Transcription factor, and just FYI, we eukaryotic cells have thousands of different transcription factors. Okay? So we don't just have one transcription factor, we have thousands of different types of transcription factor that binds to different sequence of DNA. Crazy, right? We're so complicated. But to keep it simple, we're just gonna call it transcription factor in general. So in order for eukaryotic cells to begin initiation, the first thing is first, RNA polymerase can't see it. So they're gonna need transcription factor. And these guys will be like the glow in the dark, right? It's just it's like, hey, over here, get over here. And now, your RNA polymerase is able to attach and recognize it. Okay? That'll be initiation. Okay? And then elongation, this is the same thing, is when I start creating the RNA. Okay, that's elongation. And then termination is when it cleaves off. Okay, and when this guy cleaves off. Now, I don't, in this class, I'm not going to teach you about the termination of eukaryotic cells. It's very complicated. That was my thesis project. I do not want to talk about it. It was really complicated. But just know it has to do with polyadenylation. But don't worry about that. Just know the prokaryotic termination, which is self-termination, and row-dependent termination. Okay? All right, any questions so far in regards to eukaryotic cells and prokaryotic cells transcription? It wasn't too bad, right? All right. The next one we're going to talk about now is going to be called pre-mRNA processing. Pre-mRNA processing mainly occurs in eukaryotic cells, not in prokaryotic cells, okay? So this is actually something very important in eukaryotic cells, and that's because a lot of our genes, we have something called introns and exons. And typically, introns is the one that's being removed, and exon are the one that's being expressed. So exon, express, okay? And the mechanism behind intron, exon, I'm just going to pre-mRNA processing works. You have your mRNA. So imagine this strand that I just transcribed. I enlarge it, and now it looks something like this. Okay, imagine this is the same red strand, but just got enlarged. You see that? All right. All right. So intron in this red strand, it contains introns and exons, and only the exons are the one that's being expressed. So first thing is first, in order for pre-mRNA processing to occur, two things happen. Well, three things. The first thing is going to be something called the 5' cap. 5' cap here is basically a methylated guanine. Don't need to worry about the detail, but just know it's called 5' cap. And the 5' cap, think of it like a Captain America shield. Super strong, super sturdy, almost indestructible, right? So in other words, it's really protecting this mRNA from being degraded from any enzymes or protein in the environment. So that would be the 5' cap, which is pretty important to protect the integrity of this mRNA. The second thing that goes in is something called the poly-A tail. Poly-A tail, right here, plays a big role also in protecting the RNA. However, poly-A tail consists of a bunch of adenine, bunch of A's, right? The amino acid, the nucleotide A, like a bunch of A's at the end. But the purpose of the A is not to really fully protect the RNA. It's mainly to protect the RNA for a certain extent of time. So in other words, the shorter the tail, the shorter you live. The longer the tail, the longer you live. And that's something very important because sometimes we want our certain protein to survive and be active for a certain time, but not forever, right? So giving each protein a lifespan, or each RNA, a lifespan, is very important because we don't want, let's just say, a anti-cancer gene or anti-cellular division gene to last forever, because sometimes we want our cells to divide. And what's worse, we don't want our cancer to live too long, so we want to make sure we want to have a tail that's very short, right? So having short tail or long tail plays a big role in our own homeostasis and regulation. Okay? So again, 5' cap is literally giving a Captain America shield to the RNA. And poly-A tail give you a timestamp, how long you have to live before we destroy you. Okay? And that's the second step. And the third step is something called spliceosome. Spliceosome is an enzyme. Spliceosome is an enzyme, and I'll represent that as my hand. My hand is going to be the spliceosome. Spliceosome, all it does is remove the introns and clip together and splice together the exons. You see that? So now, introns removed, And the only thing that's being expressed are the exons. And that's the function. And at this point, we're done. We just created our mRNA. Make sense? So any question about pre-mRNA processing? About the 5' cap, poly A tail, and spliceosome? All right. Now, this is for a fun fact. I don't think I have this on the exam. Maybe I do. Maybe there's one question. I'm not sure. But just in case I ask it, there's something else that I want you guys to understand. It's something called alternative splicing. Okay? There's a term here for alternative splicing, and if you were to transfer it out to university, especially UCI, they talk about alternative splicing a lot because there's this one professor, his thesis project is on alternative splicing, so he's probably going to force you guys to listen to it. But just in case, alternative splicing, all it is, imagine this spliceosome here, all it, in the poly A tail, well, don't worry about this, the spliceosome, instead of moving out the introns, maybe it decides to move out this exon instead. So now you have a whole entirely new gene. Or maybe you want to remove this exon and keep the two introns here. And then you have a whole entirely new gene. And this is something very, very common when you express receptors in, let's just say, cancers. For example, in prostate cancer, yes, if you have this, just exon, intron, is all like this, then that's great. This is awesome. You have a regular receptor, right? And you can inhibit it. However, in regards to some cancer, like breast cancer or prostate cancer and all that, maybe alternative splicing, this spliceosome decided to remove this guy and incorporate this guy right here. So now you have a receptor that does not act like a receptor. It's just always on. So this gene for cellular division is always going to be on because of alternative splicing. So in nature, alternative splicing do play its role. Maybe it gives you multiple. So one gene could create 5 different proteins that are all active and all important. But sometimes if there's a change in your diet, maybe someone's smoking or something, this could you could disrupt, due to free radicals, disrupt the spliceosome, and this spliceosome will start splicing out the wrong mechanism. So in other words, in regards to eukaryotic cells, it gets more complicated. But I don't think I need you guys to know too much. All I need you guys to know, just in case I ask you alternative splicing, just basically know that introns and exons could split around. That's alternative splicing. But in regards to pre-mRNA processing, the 3 steps: 5' cap, poly A tail, and spliceosome. All right. Any question about pre-mRNA processing? Hopefully it didn't went too much. All right. All right, and finally, but not least, we have your translation. In regards to translation, one of the most important thing in translation is going to be the tRNA. And that tRNA plays a big role. tRNA is actually an RNA. That's crazy, and it acts like an enzyme. That's pretty awesome. It's not a protein. It's an actual RNA that acts like an enzyme. And the purpose of tRNA, well, is to hold amino acids. So here's an image of a tRNA, and at the end of a tRNA, right here, this guy holds the amino acid. So amino acid could be one of the 20 natural amino acids, right? That's the main purpose of tRNA. And underneath the tRNA, we have something called an anticodon. This guy here is what recognizes the 3 codon sequence. Remember when you did translation, you did remember the DNA chart that you saw, like this guy? So here. So this down here is anticodon to recognize any of these sequences. And they will carry different amino acids. That's pretty cool, right? Now, not only that, this tRNA here has something very unique ability, and all our tRNA have that, and is unique for a reason. It's called the wobble effect. A wobble effect basically allows the codon, the anticodon, to be very flexible at the 3rd region. Why is that so important? Well, that's because imagine you're very strict, but then your last one, you could be, it doesn't matter. Well, in this case, let's just say the first 2, C and G, right, is very important. But then the third one is a wobble effect. Well, guess what? Even if there's a mutation and it hits one of this wobble effect, you still create the same amino acid, glycine, right? The third one, it doesn't matter, you still get glycine, right? Same thing here, wobble effect, third one, you still have, it doesn't matter if you change U to an A, it doesn't matter, you still have alanine, right? So the wobble effect plays a very important role, kind of a secondary protection, just in case there's a mutation that hits us, well, we're still protected due to wobble effect. But let's just say for some reason, okay, we have a wobble effect here. A U becomes an A. Oh no, we change a serine to an arginine. That is dangerous for sure. However, they both have the same function. This is polar, and this is also polar. So maybe it's not going to destroy the enzyme, but rather it slows down the enzyme or speed up the enzyme's reactivity. So it's not too bad. It doesn't obliterate us. It's still expressing a same functional protein. Okay, so that's the wobble effect. Allow you to kind of have that behavioral protection at your third codon. Okay.
Right.
So lastly, but not least, we're gonna go into the mechanism of translation. Now I'm not gonna show you guys this here yet, but I am going to talk about the beginning, and then I'm gonna show you a video. All right. The first step of translation, which is also very important, is you have your RNA here. You have your ribosome. This is your ribosome complex. You have your large ribosome complex, and then you have a small ribosomal subunit. Okay? Now, I want you guys to keep this in mind. The small ribosomal subunit is very important. It contains a cofactor. The cofactor of the small ribosomal subunit is an RNA. Crazy, right? The cofactor. So this is a protein, but within the protein, in order for it to be active, it has to be an RNA cofactor. Okay? Now, there's 3 steps involved in translation. The first step is initiation. Initiation is when it docks and binds onto your mRNA. Once it finds the AUG, the start codon, that's when it initiates. Okay? So initiation happens when it finds the mRNA and finds the AUG. Once it finds the AUG, that's when this large ribosomal subunit will come in. And this is where you begin your translation, where the RNA, the tRNA methionine will come in. Okay? Now, I'm just going to describe you the first initiation step, okay? And then the rest, I'm going to show a video. But any questions so far in regards to initiation? So again, initiation in regards to translation, small ribosomal subunit that contains coenzyme, which is RNA, will find and bind onto your mRNA. It will initiate once it finds the AUG. Once it initiates, it then brings over to the large ribosomal subunits, and which then initiate the whole entire process. Okay, that's pretty much it. Once you have this step, elongation occurs is when they're building up the amino acid, and then termination is when they find the stop codon. Okay, well, let's go over the video together.
Is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site.
All right, you guys notice that? So again, initiation occurs whenever the small ribosomal subunit, finds the AUG. Okay? Once it finds the AUG, then it's bring by the methionine and the large ribosomal subunit. So here comes methionine tRNA, and then the large ribosomal subunit will come in as well.
tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site.
All right, so this here is the initiation. So any question about initiation? All right, the next step they're going to talk about now is going to be the elongation, which is the second step. Elongation, all it is more tRNA comes in, and when this tRNA comes in, this guy will donate the amino acid to this tRNA, and then this guy will exit out. Okay, so again, a new tRNA comes in, this guy will donate its amino acid, and then it will leave after it donates. That's basically elongation, and and the whole cycle repeats.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
You see the repeat? All right, and remember this. Every new tRNA, when it comes in, every new tRNA that comes in, it will come into the A site. And then they will shift to the P site, and then here's E site. So here's how I want you guys to remember it. It's going to be EPA. E is exit. P is a growing polypeptide chain. So this is where the polypeptide is growing out from. And then A is amino acid site, just one singular amino acid. That's how I remember it. There's another word for it, of course, but that's how I remember it. So again, A site is when tRNA with just one singular amino acid comes in. P site is when the growing polypeptide chain is growing, so polypeptide site. And then E site is when they exit. Okay? That's how I remember EPA. Okay? And again, the whole mechanism elongation is basically new tRNA comes in, the old tRNA will donate the polypeptide to this guy, and then exits. That's pretty much what's going on in elongation. Okay?
These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is in...
Okay, so now we're going to talk about the termination, step 3. In regards to termination, all it is, it's pretty simple. It's once the small ribosomal subunit senses the stop codon, like UAA, for example, then UAA, it doesn't have any other tRNA. It recruits a unique tRNA. This tRNA is what we call a... It's basically a tRNA that does not have any amino acid. Okay? So once that one binds in, and there's no amino acid located on that unique tRNA, well, this guy, when it donates, it doesn't attach to any amino acid. Rather, when it donates, it's just like, oh, there's no more? I'll just float off. Because this particular tRNA, unique one, doesn't have any amino acid attached to it.
countered in the ACE.
And that TNR is what we call release factor. Okay? So when a release factor, or basically a tRNA that does not have any amino acid, comes in, and this guy's trying to donate his amino acid, there's no amino acid to bind onto, therefore it just leaves. Okay? So in summary, in regards to the termination, small ribosomal subunit senses a stop codon. Stop codon will recruit a release factor, and from there, the whole thing disrupts.
A release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
This is the last slide. One more thing I want to mention before we call it for this lecture is keep in mind that in regards to translation, a single mRNA, okay? This single mRNA, it's not just one ribosome that binds on it to translate. There could be thousands of ribosomes. Each one of those are small, large ribosomes, small and large and so forth. It could be thousands of different ribosomal RNA binding onto this one single mRNA and just translating it. You see that? So just keep that in mind that in reality, what we teach you guys is just one of them binding onto it to translate. But in the actual cells, one mRNA could have thousands of different type of ribosomes all translating proteins at once. Okay? So I want you guys to kind of recognize that. All right, so that's all the lecture I have for you guys. So what